Hello and welcome back, innovators. My name is Francesca Chen, your host for Next Breakthrough. In this episode, we interview Dr. Jia Zhao, a postdoc in the Kieffer Lab, which is part of the Department of Cellular and Physiological Sciences at UBC. The Kieffer Lab delves into the treatment of type 1 diabetes, focusing on a combination of insulin producing cells and microencapsulation devices and genetic engineering. In this episode, we discussed stem cell therapy, donor alleles, pancreatic, pregenital cells, beta cells. You will learn what all these are eventually, so don't fret. Microencapsulation, CRISPR and gene editing, hypoimmune cells, and much more. We ended off by addressing the efforts needed to make these emerging diabetes therapies more accessible and less expensive to a larger population. There are lots of fascinating components embedded in our interview. And as always, should you find anything you want to look into on your own time, please do. As well, specifically for this episode, I would recommend looking into the Yamanaka factors Dr. Zhao and I talk about a little later in this interview. So let's hop on in. Without further ado, here is Dr. Jia Zhao. Hi.
Hi, nice to meet you.
Yeah.
So the first question, I think you should start off by telling the general audience what is the fundamental concept behind using stem cells to treat diabetes, which I know is a very fundamental part of your research, and why is using stem cells such a revolutionary approach?
Let's imagine your pancreas is like a factory that produces insulin, but that the hormone that keeps your blood sugar in check. But in the case of diabetes, especially type 1 diabetes, that the factory goes on a permanent strike. So right now, people manage this by taking insulin shots, but it's like doing deliveries by hand when the factory is closed. So you can keep things running, but it's exhausting and never quite the same. And stem cell therapy aims to fix this issue at the root. And we scientists, we can take stem cells, which is a type of cells that are basically, you can think of it as like a blank state. And we can teach them to become insulin producing cells. And the way your pancreas used to do it naturally. And once we implanted the insulin producing cells into our body, these lab grown cells can sense when the blood sugar rises and release insulin in real time. It's just like switching the factory lights back on. And why is this revolutionary approach? Because it's not just managing diabetes, it's replacing the lost machinery. It's the difference between forever juggling your syringes and glucose meters versus letting your body to do what it was built to do. So the stem cell approach could move diabetes care from a constant maintenance to an actual cure.
Wow, and
Uh,
is it permanent to implement these lab-grown cells into your body? Is that a permanent way that, well, they can continue supporting this insulin creation permanently and ongoing, or is there some sort of more nuanced as to always having to implement more stem cell therapy treatment?
That's a good question. This is our ultimate goal. We hope it's a one-time or less frequent treatment rather than constant maintenance. So it's our goal to keep those stem cell-derived islets healthy and functional in our body. But there's many ways to address this issue. Yeah, it's challenging, but it's our ultimate goal.
So currently, if
Yeah.
you can talk a little bit about what can stem cell therapy do now for the patient?
Uh, you mean what kind of
Oh,
the
in terms of, sorry, I should have specified that. In terms
Mhm.
of how long can it
Ah,
support the insulin production currently?
yeah. Currently, it's still at the most of the study is still at the preclinical stage. But if you look at the recent news, like the Vertex clinical trial and Sana Technology clinical trial, actually it's relatively quite short. So for current clinical trials, only track the patient for about 1 year. But people don't know if it can survive longer. Those clinical trials are still ongoing. So we are still keep on tracking those patients We hope it could survive longer. Using donor islets, actually, they already have more than 20 years of tracking of the patients, and it terms that the medium survival of those grafts can last like 7 to 8 years, but it's unknown about the stem cell derived islets.
Oh, and then
Yeah.
this 7 to 8 years, I think you mentioned. What type of stem cell therapy is that?
Ah, what I mean, the 7 to 8 years, that's from the cadaveric donor islets. It's not stem cell derived islets.
Oh, so in
Those
the
are
donor,
different. Donor
can you
islets.
explain more about how that can also be a way to increase the insulin production in diabetes patients?
Yeah, that's actually another approach. The donor islets is from, like I mentioned, it's from the deceased cadaveric people. So it's very limited source. We do not have many donors for people to doing the islet transplantation. So that's why stem cell has the potential to as a renewable and unlimited source, alternative source to those donor islets. Um, yeah, that's why the stem cell approach is emerging recently. Yeah.
That's really interesting. Can you
Mm.
talk a little bit about how the stem cells are grown within the lab and how that becomes an effective therapy approach?
Yeah, that's very interesting, right? That's why I chose my PhD and
Hmm.
pursuing. Yeah, it's really interesting and like it's like a mystery. So coaxing the stem cells into insulin producing cells, you can think of it like guiding a student through school. So stem cells start as a blank state, as we mentioned, and then we can give them a step instructions through carefully timed addition of some small molecules, nutrients, growth factors into the culture medium, and then so they can gradually specialize into the cell types that make up a pancreas. So in short, first we can coax stem cells into let them become general gut cells. And then the pancreas progenitor cells, it's a precursor of the real pancreas that making insulin. And eventually, it can get into the insulin producing beta cells. Yeah,
And
it's
then
a
what's the
gradual.
what's the implementation process of having these well-grown stem cells in the lab and putting that actually into a patient's body? What's the
Mm,
process for that?
oh, generally, from bench to bedside actually is a really long way, and um, it's actually very different. In the lab, we're focusing on making the cells very efficiently differentiate into the insulin producing cells. And you can use like animal derived recipes or bacteria derived recipes like extracellular matrix to make the cells healthy. But if you want to translate these technologies into real patients, we wanted to remove any of those xenogenic. What I mean xenogenic is non-human source. So we need to remove any animal-derived growth factors or bacteria-derived growth factors to coax those stem cells. Because once we have using any xenogenic materials to differentiate those stem cells, and then after implantation of the stem cell derived islets into our human body, it may trigger some foreign body responses. And we don't want that. So when we translate our lab-made stem cells into the patients, actually we need to have a really dedicated facility to remove any of those animal-derived materials and find alternative source to differentiate the stem cells. You can imagine the facility is very, very clean and have a very stringent quality control to make sure the generated insulin producing cells are very clean and human body friendly.
Oh wow, this seems to be a very complicated process from,
Yeah,
as you
very
mentioned, from bench
complicated.
to bedside.
Yeah, actually, in my academic lab, we don't focus on those part. But when we translate our insulin producing cells, we need to outsource our differentiation protocol to very dedicated company to ask them to generate those GMP grade and GMP level cells, and then goes into the patient.
Wow, yeah. And
Mm.
in your work in the academic lab, how does you're, I'm guessing you would try and improve stem cell therapy and also maybe find a way to create new stem cell therapies. Is there something in that realm that we can share with us today? Maybe some things that you're doing in the lab that keeps progressing this development of stem cell therapies for diabetes patients.
What do you mean by new therapies? We are currently focusing on, maybe I can answer in this way. So to cure diabetes, to treat the diabetes, actually, you could produce several products. For example, we can produce pancreatic progenitor cells, which is a not terminally differentiated cells. It's a progenitor cell, but the manufacturing cost is low and the differentiation time is quite short compared to the eventually mature insulin producing cells. So one type of the stem cell therapy for diabetes treatment, you can use the progenitor cell, which is cost effective for many pharma companies. And the other cell type, the therapeutic cell type, would be the relatively mature insulin producing cells. So those relatively mature cells, once goes into the patient, they can quickly reverse diabetes because they can already produce insulin. But for the progenitor cells, because they're still precursor, they're still immature, if you implant those type of stem cell therapies into the patient, it requires a relatively longer time compared to the insulin producing cells to become functional and release insulin in the patient. So you indeed could have several types of stem cell therapies for the diabetes treatment.
And which one are you working on mainly at the moment? Could you elaborate maybe a little bit more on that?
Yeah. We are currently working on the mature insulin producing cells because it can, compared to the progenitor cells, it can quickly accelerate the diabetes reversal into the patient. Yeah, so we're working on that. But if you go to like 10 years ago, people focusing on the pancreatic progenitor because the science is progress gradually. So 10 years ago, people can only generate very good quality of the pancreatic progenitor cells. But after about a decade, people's continued attempt to driven the differentiation process in vitro so that people right now can produce relatively mature type of the insulin producing cells. So many of the people at current days, they are focusing on the insulin producing cells rather than the progenitor cells. Mm.
Wow, that's really cool. Wow, I didn't know that there were two types of stem cells that are currently being
Yeah,
progressed to treat
yeah,
diabetes as a whole.
yeah.
Can we talk a little bit about maybe deviate a little bit from stem cells specifically to microencapsulation devices?
Mm.
I think you guys work on that primarily on in the lab as well.
Mhm.
I understand that there's a protective layer of the microencapsulation. Why is that so crucial to successful implantation? And what happens to the cells if they don't go through this process of encapsulation?
You are talking about the macroencapsulation, right?
Yes, that's
So the
correct.
idea of the macroencapsulation is to give the implanted stem cell derived alleles a kind of, like you said, protective layer, protective bubble. So it's crucial because the immune system is extremely good at spotting anything it thinks it doesn't belong to the body, including those therapeutic cells that we implant into the body. And if we left it unprotected, it will attack and destroy our stem cell derived islet pretty quickly. And the macroencapsulation device is like a smart filter. So we want them to let in the good stuff like oxygen, nutrients that the cells need to survive, and then allow the insulin to release and get out into the bloodstream to help us lower down the blood glucose. But those devices, we also need them to block the immune attack cells, right, from getting in. So this gives the cells a fighting chance to survive and keep doing their job to control blood sugar. And why, if they encapsulate, what happens to the cells? So you can imagine, without encapsulation, you would either need a very heavy duty immune suppression drugs to inhibit your systemic immune system function, but which will also comes with many side effects. Or you would watch your implanted graft cells get rejected in a pretty short time. Yeah. So with the encapsulation device, the hope is to move toward a safer and more durable therapy, and the cells can focus on doing their job rather than survivors of the immune cell attack.
And the microencapsulation device that your lab is working on right now, are they completely foolproof? Like, or is there some sort of um, still consistently some sort of difficulty with either their implantation or how they interact with the body's immune system? And how do they protect the stem cells inside?
You mean the technical hurdles when we using this technology devices?
Yeah, so technical
Um,
hurdles, I would say, in implantation, like the actual implantation of it into the body, as well as how it interacts with the immune system while still inside the body.
Mm.
If that makes sense.
If I understand your question correctly, so uh, like you mentioned, those device, we want those devices to stay invisible to the body because those are foreign materials. And as soon as you put any foreign subject inside into the body's natural response is to protect itself by forming some scar tissue around it. So a process that we call fibrosis. So many of the technical hurdles or the interactions of the macro encapsulation devices with your body is that they they may trigger some fibrosis reaction. It's good for wound healing, but it's bad for us because those scar tissue once formed around our grafted cells, it can block oxygen and nutrients from to uh, reach your implanted insulin producing cells and eventually starving them. Yeah, so that's one of the biggest technical hurdles and what we typically see when we implant macro encapsulation devices into our body. So it's very common to see the scar formation and fibrosis. But we have some mitigation plans and some strategies to prevent that formation.
Yeah, could you
Mm.
actually, while bringing that up, could you talk a little bit about what your lab
Some
has
strategy.
devised to, yeah,
Yeah.
some strategies to tackle this issue?
Yeah. Actually, this really heavily rely on collaboration. We focusing on the stem cell therapy, but with collaboration with, for example, like a material scientist, or bioengineering scientist. So we currently focusing on two strategies and a collaboration with them. So one is that those biomaterial scientists, they can design some smarter materials. So they can use some specially designed biocompatible membranes that are less likely to trigger an immune reaction. And the second strategy is that they can engineering the membrane surface or configuration. So making the device surface, for example, ultra smooth or coating it with some immune-friendly molecules. And we try to convince, so in this way, we try to convince the body that the device is harmless, like a stealthy mode for the implants. Yeah.
And is that working well so far? Like, do you see lower amounts of fibrosis or scar tissue developing around the microencapsulation
Mm.
device with that new engineered surface?
Good question. I think it's working pretty good in animal models, but it's controversial in human patients. There are many pharma or startup company in the US, you can check. Um, Many companies, they are developing and iterating those materials for the microencapsulation devices. It's controversial because even though you could see very good results in mouse, in rodent models, but in human it's always different or complicated. Yeah, I would say this technology is still progressing, it's advancing, but I can say it's not perfect. It's still not perfect.
So it has to go through uh, more engineering, we shall say,
Mhm.
in order for it to actually be implemented within clinical trials?
Uh,
Or has it
yes,
already been implemented
good
in clinical
question.
trials and there's boundaries to
Mhm.
actually implementing it in actual
Mhm.
clinic?
Good question. So, Tim, So Dr. Tim Kieffer, he has a very good paper published a couple of years ago, published in the Cell Stem Cell. So they're working with an industry company called Veracyte. So they developed a macroencapsulation devices and they tested in human patients. So that's a clinical trial. And they show very promising results, but still they can see fibrosis formation in the patient. Yeah, it's already entry the clinical trial, but people are still optimizing the device configuration, the membrane biocompatibility. So yeah, it's promising. Mm,
That's really cool, and it's really great to know that you guys are developing something within the lab and within collaboration with other labs and other companies that can try
yes,
to foolproof, we can say this
proof of
microencapsulation device.
yes.
Oh, yeah. And maybe we should just speaking on the topic of engineering,
Mm,
let's delve a little bit into genetic engineering and especially CRISPR.
mhm.
Uh, can you walk us through a specific example of how might one use genetic engineering or CRISPR to correct a gene mutation in a patient's stem cell that causes this defect in insulin production, which leads to diabetes?
Yeah, sure. So maybe let's take a concrete example. Some rare forms of diabetes are caused by single gene mutations, such as the defects in the insulin gene. This is a very simple example. If some genes have defect in the insulin, which means that it cannot generate or makes insulin, and CRISPR is like a process editor. So we locate the exact typo in the DNA and then using the CRISPR technology to cut it out and then replace it with the correct spelling. So once the correction is made, and we can get those stem cells to become insulin producing beta cells. And because the faulty gene has already been fixed, so the cells should now respond properly to the blood sugar and make insulin. Yeah,
That's really cool. So I guess we could say it this way. So on one side, you could have stem cell implementations. You can put stem cells into a diabetes patient's body for those stem cells to produce the insulin for them. But on the other hand, you could use CRISPR to genetically engineering a mutated gene to correct it in order to fix it back into the normal state or wild type, I believe, to
yeah,
produce more insulin and reverse
yeah,
the diabetes.
yeah. That's exactly the idea. Yeah.
And then which approach do you lean more towards in your lab? Because I think you do a combination of both. Is that
A
correct?
combination, yeah, yeah, because that's the power. That's the power of the combining the CRISPR gene editing technology with the stem cells. So on the one way, you can make healthy cells to like a therapy to treat diabetes, and but on the other hand, like you mentioned, we can also generate mutations and then correct the mutations to probe their function to see what a specific gene, what's its role in diabetes development. Yeah, so we are doing both.
Really cool. And what kind of new drugs or therapeutic approaches, as we talk about those two singular approaches, are you hoping to discover or develop in your research right now? I guess we
You
could say, how are you
Mhm.
specifically hoping to advance one or both of these approaches of delivering or reversing diabetes for a patient?
Sorry, could you repeat your question?
Sorry.
Mhm.
I know we talked a lot about these two approaches for reversing diabetes in patients.
Yeah,
They've been quite developed, I would say. Currently, they're very advanced, and I know you guys are going to continually trying to advance them further. Is there
yeah.
something specific that we can take from one of these or both of these approaches that you specifically are developing right now, as a postdoc in the Kieffer Lab?
You mean the stem cell therapy? Um, what we are doing at the lab is we just generate the normal healthy insulin producing islets. And um, there's some people that are trying to generate super islets so that the islets that can generate more insulin to make them more robust to produce insulin and to reverse diabetes. But what we are now doing right now is optimizing our differentiation protocol, trying to push the insulin producing cells more functional and more responsive to glucose oscillations. Yeah, this is
Yeah,
what we are trying to do.
yeah, that did answer my
Mhm.
question.
Mhm.
I think I was mostly focusing on you as a postdoc in the Kieffer Lab. But yes, I think the Kieffer Lab's overall approach is somewhere that you also fit your research into.
Mhm. Yeah.
Thank you for that. And for I'm actually this is a curious question of my own, but the stem cells that you use to further develop the stem cell therapy, is that taken from donors?
Oh, the stem cells. That's a good question. Actually, there are several sources for the stem cells. One is called embryonic stem cell, human embryonic stem cells, which is commercial. Actually, it's very long time ago. Some very brilliant and smart scientists they took from donor very initially, and then they developed those cell lines because it's immortal, and right now they are commercial. So you can use the commercial embryonic stem cell lines, and on the other hand, you can also use induced pluripotent stem cells, which you can actually literally generate obtain them from any patients. But how can we obtain that? Because in our human body, we do not have those pluripotent stem cells anymore. Otherwise, we will form tumors in our body. So what we do is that you can take some somatic tissue, like your skin or blood or urine samples, and then we can using I'm not sure if you have heard about the Yamanaka factor, which is a Nobel Prize technology. So people can use those Yamanaka factor to reverse and reprogram those somatic cells into the blank state of the stem cells. So people can generate those stem cells in the lab as well.
Oh wow, that's really cool. I've never heard of sorry, could you repeat that? What factor was it?
Yamanaka factor, a
Yamanaka. Oh
Japanese, yeah, very famous Japanese scientist. You can Google it. Yeah.
yeah, listeners, Google that.
Mhm.
I'm
Mhm,
sure, yeah,
mhm.
I think I will too. I think there'll be lots
Mm.
of interesting information about that as
Yeah,
well.
iPSC, yeah, induced pluripotent stem cells. This is really quite commonly used at nowadays. Yeah.
Yeah, and actually, this is a good transition to, I think, looking ahead at the future of this field. So what are some, I don't know, exciting future advancements? I think we've talked about this a little bit, but let's go more in depth that your lab or you as a researcher are hoping to accomplish in the near future. And these are realistic goals that I think you're most optimistic about.
Yeah, so looking ahead, I'm optimistic about three advancements that could transform the diabetes treatment. So first of all, as we already discussed, we are getting closer to proving the clinical efficacy and safety of the stem cell derived islets. There are already several clinical trials ongoing, and it looks really promising. So clinical trials are already showing that those lab-grown insulin producing cells can restore the natural glucose control. So, which is a very important first step toward making the cell-based therapies as a real option for patients. So that's the first thing I'm super excited about it. And second, because we mentioned the immune attack from our body, right? So a second promising future direction is that we generate hypoimmune cells or immune evasive stem cell derived islets. And we can, how to achieve that? We can using CRISPR technology to engineer these cells to hide from the immune system. And we hope in this way could eliminate the need for the lifelong immunosuppression drugs. Yeah. And the third what I would think of is that my long-term vision is to move toward a deviceless approach. We talk a lot about the macroencapsulation devices, right? But I think in future, our ultimate goal is to make those devices unnecessary. So by
Mhm.
engineering, for example, by engineering the graft cells, the local environment, microenvironment, for example, improving the oxygen delivery and encouraging the rapid blood vessel growth or supporting those cell survival. So we aim to create a setting that the implanted cells can integrate and functional naturally without requiring protective devices. Yeah.
Wow, that's really very, very cool. And based on what I heard about what you guys are doing in the lab, it seems like you really are working hard towards these three goals that you just mentioned. Actually, I have a question. So the second thing you're optimistic about that you talked about, hypoimmune cells. Can we zoom in a bit about how genetic engineering can mask these cells from the body's defenses or the immune system's defenses?
Yeah, that's a good question. So, with genetic engineering, actually, we can mask these cells by tweaking some certain genes so that they no longer display their molecular ID badges that trigger an immune attack. So, in simple terms, it's like giving the cells an invisibility cloak. They can do their job of making insulin without constantly setting off the alarms in the body's defense system. Yeah. Um, to trigger an immune response, actually, your foreign cells, they have some molecular IDs in the cell surfaces, so they can be recognized by your body's immune system. And by genetic engineering, we can mask those surface molecule IDs to make them inert or just knock them out, so that your body thinks, oh, it's not foreign cells, it's just myself. So it won't trigger an immune attack. Yeah, we have those
Yeah.
strategies.
And also, correct me if I'm wrong, but the goal of the microencapsulation device, the reason why it's there is that to block the molecular ID from being recognized as a foreign stem cell by the immune system.
Yeah, you can think of this way. So macroencapsulation device is like to set a physical barrier. We
Hmm.
don't change the cell product. We don't change the cell molecular ID. So we keep them as what it is, but we have a physical barrier. But in another way, if we don't want to use the microencapsulation devices, we just implant the cells into the body and we want the cells quickly integrate into your host body, and we can use genetic engineering. And that's the concept of the hypoimmune cells. Yeah,
Oh,
so
wow.
that we can, yeah, using this technology, we can eliminate, literally, we can eliminate the need for the microencapsulation device. And that's the idea of the deviceless approach.
Wow. And do you think these hypoimmune cells could provide like a functional cure for diabetes? Do you think it'll be long lasting, um, and
We are
change the
already
current
doing that. Yeah,
already?
we
Okay,
are,
yeah.
yes, yes, we are already doing that, and also other labs. There's a company called Sana Technology, which is based on the San Diego, if I remember correctly. So they also developed the hypoimmune technology. So they are using genetic engineering to knock out some very critical molecular on the surface to make the your body don't recognize those foreign cells. There are already technologies over there, and we are also working on that. And the results, our results is still working on the preclinical animal models. It looks very promising. And the Sana technology, they already tested those hypoimmune stem cell derived beta cells into the patients, and they currently released their results about 1 month, if I remember correctly, or 3 months, 3 months. And those patients received those hypoimmune stem cell derived islets. They do not require the microencapsulation devices, and they also eliminate the need of the immunosuppression drugs.
Oh wow, that's really, really
Yeah,
good
yeah.
news. Yeah.
Yeah, yeah.
Um,
Sorry, I need to correct
yeah, go
myself.
ahead.
So, those publication actually, though they genetic engineering the donor islets. So they are using their hypoimmune technology on donor islets. But their next step plan, what I know is that they're already working on generating the hypoimmune stem cells to make hypoimmune stem cell derived islets. Those are their next step plans. What they already published is working on to generate hypoimmune donor islets. Yeah.
Yeah, and do you
Mm.
think either for the hypoimmune donor outs or the hypoimmune stem cells, do you think these therapies will, I mean, currently, clearly their development isn't cheap. It's relatively expensive, I would say. Do you think how are we going to make this therapy accessible and affordable to
Mm.
patients, rather than just a privileged few who can afford it?
That's very, very important and practical question. So, developing the stem cell-based therapies takes really significant investment, and it should not just reach, like you said, a small and privileged group. So there are several strategies that we can improve the accessibility and affordable to a wider range of patients. For example, we can develop technologies to scale up our cell product manufacturing process. So, for example, we can refine our technologies, and we are working on ways to produce the insulin producing cells more efficiently and in larger quantities. We can use bioreactors rather than using some small flasks, so that we can, in this way, we can drive the costs down over time. And second, we should simplify the surgical procedure, because if it's very burdensome, it's mentally stressful, it will also reduce the accessibility. So the clinical scientists, they are working on to make the surgical procedure much safer and minimally invasive and easy to standardize, so that many patients would more like to accept those procedures. And the third, what I can think of is that we should collaborate and work with partnerships and the policymakers. So, for example, we collaborate between academic labs and biotech companies and healthcare providers and policymakers. So, this will be very crucial to ensure the fair pricing models, because if it's too expensive, that would be not practical for low-income countries, right? And finally, I would say the global equity is also very important. We need to plan for accessibility beyond high-income countries, so the therapies benefit people living with diabetes worldwide, not just in select groups.
Yeah, I think that's a really good layout of the plan towards the goals that we eventually want to reach. And I just want to add, it's really inspirational, I think, to hear about like it's not just one lab who is solely trying to cure diabetes, who is the only one that is putting in their effort and time in order to make these STEM therapy that we want to reach. It's a whole collaboration of many different labs across many different countries, or even people, those in hospitals that are all collaborating to make this happen.
Yes, yes, that's correct. It's really a collaboration thing. Yeah.
Mhm. Yeah.
Mhm.
And on that note, I think that's yeah, I think that's a really good note to end our interview off with. Just knowing for those at home who know someone or have diabetes that there's so many efforts and successful so far that are trying to um, implement ways that we can treat diabetes. Oh,
Mhm.
yes.
Yeah, I'm
Thank you
glad.
very much. Yeah, go ahead.
Yeah. I'm very glad. I find your questions really great.
Thank you very much. And I find your answers really very complex. And I learned a lot from this actually, and it's
Thanks.
really
Thanks.
inspiring. So
Yeah. Thank you for having me again.
thank you, Dr. Zhao.
Yeah. Yeah.
Have a great rest of the day.
Yeah, you too. Have a good evening.
Yeah, you
Bye.
too. Bye.